Hello everyone, I would like to welcome you to the QIAGEN webinar on Sample to Insight at LightSpeed, a barrier-breaking workflow for inherited disease NGS testing. My name is Marianne, I'm the Global Campaign Manager for QIAGEN Digital Insights, and I'll be the host and moderator for today's webinar. So first, a few housekeeping points. During today's session, all attendees will be in listen-only mode to avoid any background noise during the presentation. In our webinar window, you'll see several panels on your browser. There is a media player window that will be showing our presentations. Depending on the size of your screen, I would encourage you to play around with the sizes of the windows if anything is unclear or too small. You can minimize or maximize the window using the icons in the top right corners, or you can also enlarge it by clicking and dragging the edges. On the webinar screen, you'll also see a Q&A section called Ask Question. If you have any questions for the presenters or regarding our solutions in general, please let us know here. We will address questions during the Q&A at the end of the presentation, and if there are any questions we don't get to today, we will follow up with you by email. Now, I'd like to take a minute to introduce our speakers for today, Krishna Amin, Dr. Leif Schalzer, and Dr. Georgios Stamoulis. Krishna Amin has worked with various technology developmental groups for qPCR and next-generation sequencing technologies. Currently, she leads the targeted DNA-seq portfolio at QIAGEN. Dr. Leif Schalzer has worked with many aspects of bioinformatics, ranging from microbial genomics and metagenomics to biomedical applications. And Dr. Schalzer is now the Director of Global Product Management for QIAGEN Digital Insights. Dr. Georgios Stamoulis is a molecular geneticist with more than 12 years of experience in human genetics and genomic diagnostics and research, and is the Director of Global Product Management for Hereditary Diseases at QIAGEN Digital Insights. Now I will hand the presentation over to Krishna.
Thank you for the introduction. Before we get started, I would like to start with our legal disclaimer. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, see the respective QIAGEN kit instructions for use or user operator manual. QIAGEN instructions for use and user manuals are available at www.qiagen.com or can be requested from QIAGEN Technical Services or your local distributor. All exome sequencing aims to screen for as many disease-relevant loci as possible, and Human Gene Mutation Database, or HGMD, attempts to collate all known or published gene lesions responsible for the human inherited diseases. In the next slide, we'll look at how well these disease-causing loci are covered in different exome panels. We will particularly look at disease-causing mutations and likely disease-causing mutations. Both of these variant classes represent greater than 96% of all active HGMD mutations. So we start by looking at different exome panels, with first looking at the percent of HGMD mutations that are covered by each of these panels. And as you can see, the QIAGEN human exome covers 99 or greater than 95% of all HGMD mutations, even with its reduced ROI size. While there are other suppliers like supplier I and supplier K that cover greater HGMD mutations, their ROI size is greatly increased, requiring more sequencing capacity. We also looked at what fraction of HGMD loci would be covered, sufficiently covered or sufficiently enriched to allow variant calling at greater than or equal to 20x coverage. And at an average of 60x coverage, we see that QIAGEN human exome is able to cover 91.5% of HGMD mutations. Supplier K here did cover 1% more HGMD mutation, but at the cost of 26% more sequencing. Other suppliers like supplier X, T, and S, even with their large ROI size, we're only able to cover about 87% of HGMD mutation. So this makes the QIAseq the most compact whole exome solution on the market, while covering the most disease-relevant loci and reduce sequencing cost. So next, we wanted to look at how do we manage to target even more HGMD loci, and how do we get ourselves to 100% covering HGMD mutations while keeping the target region small to really efficiently sequence and reduce your sequencing burden. So as of today, there are more than 6,000 monogenic disorders with known molecular bases linked to far more than 4,000 genes. And there's even greater number of genes that are involved in complex diseases. HGMD currently lists more than 13,000 disease-related genes, and 9,000 of them are clearly associated or likely associated with an inherited disease. So targeting coding sequences of all of these 13,000 genes would result in a relatively large target regions. This does not enable high target coverage at a reasonable sequencing cost. On the other hand, small clinical exome panels only focuses on about 4,500 genes with clear association to monogenic disorders. This does save the sequencing cost, but it fails to detect a large number of disease-related variants. So this is why we've developed an actionable exome panel to detect more pathogenic variants with even more reduced efforts. So QIAseq Actionable Exome Panel covers 100% of the HGMD mutations with just 12.3 megabases of ROI. And we're able to cover all 100% of these HGMD mutations at an average 60x coverage with QIAseq Actionable Exome Panel. As I mentioned, the X-type panel targets all or 100% disease-causing and likely disease-causing variants listed in HGMD. There are more than 300,000 variants that are listed in HGMD that include the variants in regulatory regions, splice sites, as well as deep intronic variants. Due to our proprietary bimodal design, target size for the actionable exome is reduced to just about 12 megabases, while still targeting more than 10,000 genes. More than 4,300 genes comprise a high number of disease-causing variants and will be enriched entirely. This includes whole coding sequence and 10 base pair splice regions. More than 6,000 genes with a low number of disease-related variants are enriched partially, but selectively to include known disease-relevant variant loci in exons, splice regions, introns, as well as regulatory regions. So even with its reduced size, QIAseq X-type actionable exome is a highly suited solution for rare disease detection. Let's review the QIAseq X-type technology for target enrichment for exome or actionable exome sequencing. First, you start with your genomic DNA and build a whole genome shotgun library. Next, you perform probe hybridization where the probes would bind to the regions of interest. The QIAseq X-type hybridization capture technology uses double-stranded DNA probes. This enables the capture of both strands of your DNA library. It also helps generate higher complexity library even when you're using a smaller input amount of your DNA. After that, you use a streptowide-encoded magnetic beads to pull down the probes that are bound to your regions of interest. And after washing away all your unbound library fragments, you're left with your target-enriched library that can undergo PCR amplification and QC before sequencing. Now, it is well known that during the hybridization step, the efficiency of probe binding can be influenced by the GC content of the target regions. And sequences with extreme GC content, either very high or very low, can reduce the capture efficiency, and this results in an uneven coverage across your targeted regions. With double-stranded DNA probe in a highly optimized workflow, QIAseq X-type technology minimizes this GC-induced bias compared to other hybrid capture-based solutions in the market. So here, you can see a nice horizontal distribution or uniform distribution of your captured regions with X-type actionable exome with 8-plex hybrid capture reaction with genomic DNA compared to some of the other suppliers. You can also see the distribution of the captured region is consistently uniform even when we're working with a highly fragmented sample such as cfDNA. And we know that the insufficient enrichment of this extreme GC content can lead to failure of detection for many disease-relevant genes. So for example, here we look at ADAT3 genes, mutations in this gene are responsible for the autosomal recessive intellectual disability. And the average content for average GC content for this gene is greater than 70%, which could impair the probe hybridization. So here you can see that supplier I is not able to provide a uniform coverage. In fact, you see a drop in coverage across this gene. And with supplier S, you can see a highly heterogeneous coverage of the gene. On the other hand, together with supplier K, QIAGEN provides a homogeneous coverage of this entire gene. So QIAGEN provides even coverage of the targets across a wide range of GC content to ensure minimal dropout and improving the capture efficiency as well as the specificity. So one advantage of the QIAGEN X-Hib technology is that it offers flexible hybridization time. Here we looked at 30-minute hybridization, 2 hours of hybridization, and overnight hybridization of the probes to the targeted regions and their impact on the target-based coverage. And as you can see, we see relatively high coverage uniformity with different hybridization times. In fact, overnight hybridization time made no difference to the percent bases covered at 20x coverage. Another advantage of the QIAGEN X-Hib technology is the flexibility of multiplexing different hypercapture reactions. So we looked at 1-plex actionable exome versus 8-plex actionable exome hybridization, as well as the 8-plex human exome hybridization. And again, we see up to 99.5% target bases that are covered at 20x or greater coverage, regardless of single or multiplex hybridization and capture reactions. Besides the genomic DNA, also formalin-compromised DNA and cfDNA are also fully compatible with the X-Hib panels, particularly with the X-Hib actionable exome panel. Here we see when we're using challenging samples such as FFPE and cfDNA, we retain the sufficient coverage of disease-relevant HGMD loci. We also measured the duplication rates across different cfDNA input amounts. While the duplication rate does increase with increasing cfDNA input amount, it is still relatively low to enable variant calling with high confidence. So this just shows that due to the compact size of the X-Hib actionable exome panel, sequencing at greater depth still remains affordable even with challenging samples like FFPE DNA samples or cfDNA samples, and we're still able to retain a comprehensive coverage of all disease-relevant HGMD loci. Now let's look at the complete NGS workflow for the QIAGEN X-Hib actionable exome. The QIAGEN X-Hib technology uses a universal, fast, and fully automatable workflow. The first step in the workflow is library preparation of your DNA to build a whole genome shotgun library. Using a QIAGEN FXDNA library, your genomic DNA is fragmented and repaired an A-tail during an FX reaction. You can also use QIAGEN ultra-low input library kit if you're working with cfDNA or highly fragmented DNA samples, which skips the fragmentation steps and goes directly into end repair and A-tailing. Following this step, platform-specific adapters are ligated to both ends of your DNA fragment. Following a bead cleanup step, a high-fidelity PCR reaction is performed to ensure maximum yields from a limited amount of starting material. Then a bead cleanup step is performed for a reaction cleanup and removal of adapter dimers. Now we come to the second step of the workflow, which is library enrichment. During the QIAGEN X-Hib library enrichment, your index libraries can be pulled up to 8-plex hybridization reaction and then dried down. During the hybridization, fragments are bound to biotinylated double-stranded DNA capture probes with a highly flexible hybridization times, ranging from 30 minutes to 4 hours to even overnight incubation. Your bound fragments are then immobilized on the step-dividing coated beads, and non-fragmented or non-targeted fragments are washed away. Enriched library fragments are then amplified using a proprietary post-capture amplification mix that allows amplification of your DNA regions with vastly different GC content. This also minimizes sequencing bias caused by PCR. Ultimately, a bead cleanup is performed to purify an enriched library that is ready for sequencing and downstream analysis. I will now hand over to my colleague Leif, who will walk you through secondary analysis through CLC Genomics Workbench.
Thank you, Krishna. For analysis of inherited diseases, the CLC Genomics platform is a preferred choice of analysis as it offers all bioinformatic capabilities needed. In addition to analysis tool and workflows for de novo assembly, resequencing, RNA-seq, microbiome, single-cell epigenomics, immune repertoire, and many other applications, it now offers ultra-fast, FastQ to VCF processing pipelines. The CLC Genomics Workbench features a user-friendly interface, rich visualizations and workflow editors, and it is ISO quality certified and works on most platforms, both in terms of operation systems and NGS sequencing technologies. With the server and cloud software, it scales to high throughput scenarios, and in combination with its versatility, it is a favorite analysis tool in many core sequencing labs. The software is fully documented and supported by a team of PhD-level scientists. Finally, it is good manufacturing practice or GMP ready, complete with audit log, user management, file provenance, and history information on all output files. Shifting gears to today's topics, I would like to introduce the new CLC LightSpeed module. LightSpeed is an ultra-fast read mapping and variant coding pipeline based on new technology consisting of optimized data structures and algorithms. We launched this module in January this year with a hereditary pipeline and will follow up with a somatic pipeline in August. We have tested it on data from various NGS sequences making use of paired-end short reads, including Illumina, MGI, Element, and Singular. LightSpeed works on any diploid species. It is purely software, CPU only, meaning that no special hardware is needed for execution. In other words, it will run on any existing computer in your lab. The optimized data structures and algorithms ensure fast and memory efficient execution, which also translates to cost savings. Its performance is competitive when benchmarked on genome in a bottle datasets. There is no downsampling involved, ensuring reproducible results. And because it is so fast and runs on generic hardware, it has a very low energy consumption and associated carbon footprint. Our beta testers included some of the largest commercial clinical test providers, and they were very happy with the results and the impact this will have on their turnaround times. LightSpeed is the world's fastest and cheapest to run FastQ to VCF whole genome sequencing pipeline available. We benchmarked publicly available 30x to 35x coverage whole genome sequencing samples from recent FDA challenges. For this, we used Amazon hardware, as this allowed full control over access to the hardware and enabled apple-to-apple comparison with data from other vendors. Here we show that LightSpeed takes 20 minutes CPU time in AWS compared with 30 minutes and 25 minutes using Dragon and Parabricks respectively. Turning to costs, this translates to 40 cents Amazon costs compared to $7 and $4 for Dragon and Parabricks respectively. To show the flexibility of deployment of LightSpeed, we tried Amazon, local workstation, and even our laptops. To our knowledge, LightSpeed is the only software which can crunch a 30x whole genome sequencing dataset on such a breadth of hardware within reasonable execution times. For 50x whole exome sequencing datasets, the numbers become even more impressive. 1 to 2 minutes execution times and a few cents of associated AWS costs. Dragon takes 9 minutes and costs $2 for whole exome sequencing. We benchmarked the quality of the VCFs on several samples, references, and sequences and achieved above 99% F1 scores, showing that not only is LightSpeed fast and cheap to run, but it also supports multiple platforms and does not compromise on accuracy. On real-world samples from collaborators, LightSpeed crunches 1,000 whole exome sequencing samples in a few hours on existing hardware, a standard high-performance computing cluster of 20 computers. The most obvious use case is in neonatal screening, where every minute can make a difference to the outcome. We had collaborators perform this retrospective analysis of whole genome sequencing data from 4 trios. All confirmed variants were identified in record time, including interpretation using QIAGEN Clinical Insight Interpret in under an hour. The CLC platform may not be the format your laboratory prefers to set up secondary analysis, as it involves local installation and efforts for getting familiar with the software. Instead, many laboratories wish to consume software as a service, and to accommodate this wish, we offer a service called QIAGEN Clinical Insights, or QCI Secondary Analysis, which is a cloud-based NGS secondary analysis service. QCI secondary analysis can be set up to accommodate analysis of NGS data for any panel by any vendor. So it is fully flexible. It is easily customized, and as this is a cloud offering, there is no local hardware required other than the computer to launch the analysis. Finally, you get access to worldwide and world-class bioinformatics and NGS expertise. The way it works is shown in this diagram. A so-called connector software is scanning for new sequencing files to appear in a specified folder on a computer near the sequencing machine, and then automatically uploads the FASTQs to the service where the analysis is kicked off. Results can be downloaded, and VCFs can be passed on to QCI Interpret for generation of the final clinical report. Now I will hand over to Georgios, who will speak about what QCI Interpret is about. Thank you for now. Georgios, over to you.
Thank you much, Live. Hi to everyone, from myself as well. My name is Georgios Tamoulis. I'm a molecular geneticist by training and the director of global product management for hereditary disease in QIAGEN Digital Insights team. So I'll jump now to the third part of our webinar, where it's about interpretation part, and we'll try to shed light on hereditary disorders with QCI Interpret. So I'll start with a quick introduction of the current landscape on the hereditary disorders. In ancient Greece, you know that Hippocrates had the theory of 4 humors: black bile, yellow bile, blood, and phlegm. And nowadays, there is an equivalent of the 4 nucleotides, which determine our health, disease, and disease status as well. The hereditary testing field evolution has been evolved through the years, and nowadays we can say that we are in the era where things are shifting towards whole exome, whole genome, from multi-gene and exome panels as the actionable exome that Krishna described before. So the vision and we think that in the near future we will have a more inclusive approach with multi-omics approach, where we can incorporate more verticals of information in order to provide more accurate diagnosis. The rise of next generation sequencing era is here, and we can see that every day we learn new things, we discover new genes. Here you can see a screenshot from OMIM on June 2nd. The genes that we know that are involved in single gene disorders are more than 4,400, and it is estimated that approximately 1 new gene is discovered every day. So that means that there is a big number of genes still to be discovered, and this is estimated around 9,000. And the interpretation process is a process that is involved through the life cycle of a human, as you can see here in this very descriptive figure, from zygote to adult. The interpretation part is extremely important. Either it is about germline testing or somatic testing in adulthood for cancer, or in germline for infancy, childhood, and adolescence testing, as well as with polygenic score and non-invasive prenatal testing. But here we will focus more about the interpretation around exomes and the actionable exome that Christian presented. So the question that arises is how someone can interpret accurately and confidently and analyze variants with more than 25,000 or 3 million variants when it comes to the whole genome. There are many challenges for clinical labs which are conducting NGS testing, such as the interpretation time. They all require a fast interpretation time and consequently a fast turnaround time. Moreover, to develop and operate proprietary annotation solutions is much costly. And also, a very challenging task is to make sure that someone can keep up with the constantly updated information on the bibliography and literature. And speaking about challenges, there are, of course, many more challenges when we look on an exome scale, where data is much bigger. So a huge amount of data means more interpretation effort. The interpretation time also can increase due to the amount of data. There is an increased demand for new evidence of accurate interpretation, as well as periodic reanalysis of undiagnosed cases, CNV analysis. Also, the fact that many diseases have high genetic and phenotypic heterogeneity for specific groups of diseases can be challenging. And last but not least, the inclusion of artificial intelligence and also the manually created content where people are trying to find an equilibrium between these two levels of information regarding the interpretation part. And I will also speak about that in a bit. I will try to give you a brief overview of QIAGEN Digital Insights. The team consists of hundreds of scientists and engineers and other specialists who work daily to keep on top of the new clinical literature and to provide products that help translate genomic data into clinical insights. QIAGEN Digital Insights provides a variety of products in different areas, from academic research on human samples all the way to clinical laboratory testing. And one of the fundamental and core things we do is to curate this evidence from the scientific literature and to keep it up to date for you. So you can get a constant current view of what's known to be clinically relevant and gain insights into your data and clinical results. QIAGEN Digital Insights is serving more than 90,000 customers worldwide. Our clinical decision support tools have been cited in over 35,000 publications. So I will move now to more details about our knowledge base, which is, I think, the most important part and what makes our interpretation workflow the preferred workflow for clinical purposes. So it is characterized by 5 pillars: depth, breadth, quality, timeliness, and context, with more than 20 million curated findings and more than 50 public databases in the QCI Interpret tool, as well as HGMD, which is the gold standard tool for germline variation. As well, the breadth of information is characterized by more than 20 years of expert curation, manual creation from our PhD scientists. The quality of this information is ensured since on this content, more than 200 PhD scientists are working and are curating manually content every day. The timeliness of this is ensured by the fact that this knowledge base is updated weekly. Some parts as well are updated monthly and quarterly, but the vast majority of the information is updated weekly. More than 5,000 variants are added daily in this context. And as I mentioned, we have more than 35,000 publications already cited and for this tool. This knowledge base is fundamental and core to our products for a reliable and fast interpretation process. And I would like to touch a little on our curation process. Our curation is sort of like a factory-like process on a high scale. We have hundreds of PhD scientists working on the curation systematically through curation protocols and processes that are rigorously tested and defined. And that provides very high quality computable evidence, which then can be used to put the data that is generated from your NGS runs into context of what's currently known about the variants and mutations detected. Evidences are created from a variety of different sources, such as scientific literature, but we also create medical guidelines like practice guidelines from a variety of sources, drug labels, clinical trials, as well as molecular data sets, population-wide data sets regarding genomic variation and potential pathogenicity of the variants. So a wide variety of different content sources that are clinically relevant is created daily. And this is done through a distributed workforce of scientists who are trained on the various protocols we use and read the literature, or review the other sources and integrate into what we call the QIAGEN knowledge base. This enables our application, including the QCI interpret, to compute and combine all the relevant information related to the variant of interest, and then put it into biological context for you. So, what are the most important features that laboratories are looking for? Is quality, speed, or cost? If you could just pick one of those, some people they may choose quality, some speed or cost. And when it comes to the interpretation part, directly to report, we make sure that we ensure that all these three aspects, quality, speed, and cost, are ensured in order to save you time, to save you money, and of course, to save lives of the patients. And now, if we look on the full workflow, the full workflow starts with a panel, a chemistry, like the one that Krishna described before, the QIAGEN actionable exome in this case. Of course, any other assay can be used. And then we have the secondary analysis part, which was described by Life. Now with LightSpeed is one of the fastest workflow and the most cost efficient. And then we have the part of the interpretation with QCI interpret, where the input that is needed is a VCF file, which can be given directly from the secondary part to the tertiary analysis part, and then move forward with the interpretation workflow. The VCF also can be done, the VCF part and the interpretation part, excuse me, can be done also through API integration in our system, and through API calls if there is a need from the lab. QCI Interpret is a platform which can be used for all hereditary applications, from hereditary cancer, carrier screening, familial variant analysis and tree analysis, whole genome, whole exome, inherited disorders, and of course somatic oncology. And one of these aspects is what Krishna presented before, the actionable exome. So QIAGEN Clinical Insights Interpret for hereditary diseases. Here we saw that is powered by our knowledge base. As I mentioned before, it provides all the interactive filters to prioritize variants and proprietary algorithms to dynamically and transparently compute ACMG and AMP variant classifications. It is characterized by a very high quality phenotype-driven ranking, which I will show you in a bit. And of course, it generates evidence-based reports, which are all based on manually curated evidence from literature, which saves you a tremendous amount of time from the interpretation workflow. And now this content of manually curated evidences is further enhanced by artificial intelligence content, which we extract from literature. And I will show you in a bit, which will be available in our upcoming release. QCI Interpret is not just like any other interpretation platform of the market. It provides, apart from content, and all the necessary workflow that someone needs to do a very accurate and reliable interpretation. All the available features and tools for the most comprehensive interpretation workflow are available, such as full transparency on the article source of all evidences, the frequent updates of expertly curated information, the phenotype-driven ranking, the AI and NLP variant citations on top of the manually curated content, as well as scalable workflow support with the API integration I mentioned earlier. And of course, the gene variant and gene disease contextual information with all the different filtering capabilities. Here, you can see I will show you some of the features that are characterizing our platform. You see that we have all the relative interactive filters that someone can use, for example, on the confidence of the variants, the frequency of variants, the deleteriousness of the variants, and many, many more filters when it comes to quality, when it comes to the type of variants, which you can adjust based on your needs. So in this way, someone can narrow down a very big list of variants. In this case, we have more than 5 million variants down to very few. And then by adding also the phenotype of the patient, this list can go even smaller and makes the interpretation even faster. Here's the phenotype-driven ranking. Someone can add the phenotypes of the patient and then dramatically decrease the list of variants down to very few. And thus the interpretation workflow is very, very simple and very fast, as well as the clinical validity feature, where we provide gene disease association strengths, which are computed for all genes. We provide also these type of diagrams where someone can see the candidate disease evidence and findings, which are associated with literature references and are available for review. And this setting of clinical validity is based on the latest ClinGen's recommendations. Moreover, we provide all the necessary tools for CNV interpretation with all the CNV literature content to be available when it comes to CNVs. And this is an ongoing effort, which is further expanded. New ACMG CNV guidelines are also included, and of course, links for external CNV databases, which are extremely helpful in order to prioritize CNVs. Moreover, on our latest release, our content has been further enhanced for carrier screening, and now we have 100% full coverage of manually created content on the ACMG tier 3 and tier 4 genes when it comes to the carrier screening as suggested by the ACMG guidelines. Moreover, we implemented in our latest release faster workflow where someone can interpret and make assessments directly from the variant list without the need to go into variant details. For example, here you see the triage mode that we have implemented. The user can assess directly from the variant list the variant without the need to look into further details, especially when it comes to variants that they are seeing often or in variants that they see that they know that are most likely pathogenic. Moreover, we have added extra information such as the ClinVar submissions, which is extremely useful in order someone to get a better understanding and feeling about the potential pathogenicity of the variants, as well as the publications which are related to its variant. And in this way, we provide more context regarding the pathogenicity of the variant and enabling the users to have a much faster decision regarding its variant. Additionally, we have managed to create a tool where the user can add all the related sample or run QC reports, specimen data, or LIMS integration, or LIMS link out, with any document that can be related to this patient, and all of them can be found in a specific folder under the specific case. Of course, when it comes to the last part, which is the report, our reports are fully customizable, and the users can add different information there, such as the ACMG guidelines. Here in this example, I have added a report from the QIAGEN actionable exome, where 2 variants were found as pathogenic. And here is an example of a carrier screening report where we provide extra information for specific diseases related to carrier screening. Here indicatively you see cystic fibrosis, as well as different information about the frequency in different ethnic groups. Now I'll speak a little bit about the future of QCI Interpret and the streamlined variant interpretation and what comes in the upcoming release. So in the upcoming release, we have now an improved phenotype-driven ranking, which becomes more variant-specific, and by taking into account much more information on the variant level, and thus we further improve the ranking of the variants when it comes to this approach, where the users are adding extra information regarding the phenotype. Moreover, now we are further giving more transparency on ACMG triggering for specific criteria. For example, in PVS1, we will be giving extra notices for the different subcases of PVS1 triggering in order the user to feel confident about this triggering and also to have full transparency and investigate further where there is a need. Additionally, we are now in the upcoming release, we will have main select and main plus clinical transcripts as default transcripts in our workflow. So the users will have now the consensus between RefSeq and Ensemble, which is the main select. Of course, the users can select different other transcripts for a gene and see these transcripts in this graph here that we have, and then they can better understand the consequences of the variant in regards to the position, if it's close to a splice site or if it's something that is needs to be further investigated. Moreover, as I mentioned, the AI content now is a new feature in the upcoming release. We are enhancing our manually curated content with AI extracted information from bibliography with full transparency on the article source of the content. This is enhanced content, a list of evidences, which will facilitate the interpretation process and consequently reduce the turnaround time. Here is an indicative example. You will have now two new icons regarding the source of literature. Here is some literature that you see. We have now these new two icons. The blue will indicate the super high confidence. The yellow will indicate high to medium confidence. And now we are eliminating any AI content information which is predicted to be of low relevance. And this is the big difference with our AI approach and other AI approaches for bibliography that exist out there. Since we do not provide you with information that we think is irrelevant and will just take much more time for you to spot what is really meaningful among all these plethora of information when it comes to AI. So we provide you only the very high quality information of AI. Here indicatively is an example. We have now all the relevant literature for this specific variant in this gene, and you can see that we have three articles which are AI extracted from bibliography with and characterized by very high quality, since the color there seems to be blue. Moreover, we are adding also another tool which for users and which can be clicked, and then the literature can be further filtered, and you keep only the literature which is related to your phenotype or to the phenotype that you gave as input to the software and the symptoms of your patients. And in this way, you can focus only on literature which is related to your specific phenotypes. You see also that in every bibliography, in every reference, we give also the context, which is extremely useful for you to easily spot check what may be more important for you. Last but not least, the feature that is coming is the automatic periodic reanalysis and alerting for new evidences in order to address one of the biggest bottlenecks of exome and genome interpretation and to increase the diagnostic yield. So far, we are providing this feature for evidences when it comes to variants that the users have been previously assessing. So for variants that have been previously assessed, we provide already this feature. Now we are expanding this feature into every single variant that you have seen in your accounts. In this way, you will be able to check periodically without the need from your side to spend extra time. You will receive automatic notifications if there are any classification changes regarding to specific variants. And then you can go back to your tests and check this information if this is relevant to you or not. And now I will switch quickly to a quick overview of the QCI Interpret. So here you can see our clinical insight, the QCI Interpret platform, where it's used for the interpretation. So real life, you can see now how what I presented you before is working. We have a list of variants here, 450 variants. Those are coming from an actionable exome-like case. And you can see that different variants have different pathogenicities, the different colors. And there are different ways that someone can go through this information in order to minimize the list. From thousands of variants, now we have 450 because we have applied basic filters that most of people apply about frequencies, about predictions, etc. So now I would like to show you the phenotype-driven ranking workflow. How is it working? So in this case, we can add different phenotypes about this patient that are available. I've already added a couple of phenotypes for this patient. I will show you how it's done. In this case, there is a description that there is a disorder of hair follicles and lack of hair follicle, and then it was also congenital ichthyosiform erythroderma, and we can add more if we want. I have already added these here for you so you can see. These are the 3 phenotypes that I've added in this case, and by adding these phenotypes, we end up with a much shorter list, only 40 variants, and sorted by the most relevant to be related to this phenotype. It seems that 2 variants, which seems to be heterozygous as well, are found in this patient, one pathogenic and one likely pathogenic. We can look more details about this specific variant, about the triggering, full transparency, what has happened, different variant details, different information on the sample, on the effect on the protein with main select transcripts pre-selected, as I mentioned, and much more information in order to give you more insights. More importantly, we have the bibliography section for this specific variant, where you can see that now we have information related to this variant. Here you can see the AI-driven information. We have now also papers coming from AI, apart from the manually curated content. Here is the information about the different types of bibliography we provide. And of course, someone can click here and focus only on what really matters. And you see by clicking here, show phenotype related references only. Now we focus only to the information and references and bibliography, which is related to the information we gave before. The congenital ichthyosiform erythroderma, loss of hair, as well as the Netherton syndrome, which was the initial disease that you can see here before adding the phenotype. For these 2 variants is the Netherton syndrome. Then by adding the phenotype information, we can further pin down more articles. Of course, there is also the information that I would like to mention here about ClinVar, where someone can click and get much more insights regarding the submissions in ClinVar, as well as the number of publications, and of course, HGMD information for every single variant. With this, now someone can take this variant and report it very easily if need be, and then create a fully customizable report, as I showed you before, with all the type of information that your lab would like to integrate in the report. So with this, I will stop live demo information, and I will switch to my summary. So to summarize, QCI Interpret is a scalable, standardized, and streamlined NGS interpretation and reporting workflow, which can interpret different types of genetic variation, including CNVs, with the most up-to-date integrated evidences, with efficient variant filtering strategies and all the available tools, as well as the phenotype-driven ranking that I just showed you. All the relevant clinical insights for the actionable exome from QIAGEN, as well as fully compatible with the LightSpeed technology for the secondary analysis. Is fully integrated in any sequencer and secondary also agnostic. It can provide any information about even if it comes to single sample analysis or to multi-sample analysis or familial variant analysis with a fully customized report. And of course, it is reducing the interpretation time by all these efficient workflows, tools, and features in order then to, of course, have a reduced turnaround time to the patients as well, and faster diagnosis for the patients. With this, I would like to thank you very much for your attention from my side as well, and we will be happy to answer any of your questions. Thank you very much.